My lab is interested in the mechanisms implicated in pro-inflammatory diseases, including necrotizing enterocolitis, and how such induced pathogenic inflammation can be mitigated to limit tissue damage. I am here today to briefly discuss our results published by the Journal of Clinical Investigation. Despite major achievements in medicine, the rate of preterm birth has not decreased and remains one of the leading global causes of infant mortality. Maternal pathogen-induced inflammation may contribute to both preterm birth and fetal inflammation. Data demonstrate that such induced detrimental inflammatory responses significantly modify the gut microbiota of neonates, dysfunctioning their gut homeostasis, all of which potentially manifest in inflammatory bowel necrosis, NEC, especially in preterm infants with risk factors. So far, the scientific premise suggests that human breast milk and the beneficial bacteria within significantly influence both the gut microbiota and intestinal immunity of the neonates, thus affecting disease risk later in life. However, we still need to explore the mechanisms involved to better understand the role of human breast milk and its beneficial bacteria, which control innate and T-cell immunity, specifically Th17 cells critically involved in mucosal protection and the pathogenesis of severe inflammatory diseases, including NEC. In the current study, we investigated the status of the microbiota of two discrete cohorts: preterm infants fed human breast milk and preterm infants fed formula. Our data demonstrate clear differences in the diversity of the gut microbiota from breast milk fed preterm infants compared to that of formula-fed preterm infants, with the Propionibacterium species heavily contributing to these differences. With an isolated, characterized, and cultured a novel Propionibacterium strain that was present in the breast milk-fed infants, which was designated as Propionibacterium University of Florida, or PFU1. To explore the potential differences induced by microbiota of breastfed and formula-fed preterm infants, we transplanted the fecal microbiota of these two different cohorts into germ-free mice. Here, breast milk-fed preterm infants' microbiota specifically increased Th17 and Treg cells, a trend that was less evident in mice transphonated with formula-fed preterm infants' microbiota. We then demonstrated that Th17 cell differentiation was dependent on bacterial dehydrolipoamide acyltransferase, DLAT, in germ-free mice monoassociated with PFU1, as deletion of DLAT gene from PFU1 chromosome resulted in ablation of the Th17 cells in these mice. This DLAT-specific Th17 cells swiftly resolved intestinal Listeria infection in mice that were fed with PFU1. Additionally, PFU1 also sustained the gut microbiota and induced critical metabolites such as citrulline involved in the bacterial pathogen clearance. Lastly, to demonstrate the impact of PFU1 in controlling neck-like injury in newborn mice, we fed pregnant mice with PFU1. The newborn mice from pregnant dams were then gouged daily with PFU1. Data demonstrate newborn mice receiving PFU1 compared with control mice significantly resisted the exacerbation of neck-like injury marked by regulating phagocytes, Th17 and Treg cells, limiting intestinal tissue damage. In summary, our studies demonstrate the critical role of human breast milk in shaping the gut microbiota by increasing their bifidogenic-like properties, influencing gut homeostasis to limit inflammation that could otherwise manifest in fetal pro-inflammatory diseases, including NEC. Additionally, elucidating the underlying translational mechanisms may lead to the establishment of less costly and non-invasive therapies for intestinal disorders, potentially by providing human breast milk supplemented with beneficial bacteria like PFU1 to preterm infants with risk factors.